Your digestive tract represents the most expansive physical interface between your internal physiology and the outside world. This biological frontier manages a rigorous immunological defense that serves as the primary gateway to your internal systems. This entire frontier is defended by a structure that is shockingly thin. A continuous single layer of specialized cells called enterocytes is all that stands between daily survival and a systemic infection. To prevent toxins slipping through, enterocytes are bolted by tight junctions. Interlocking proteins, Claudin and Occludin, function like biological teeth, zipping the gap shut. Managing paracellular permeability, they selectively open to allow nutrients into your bloodstream while immediately snapping shut to repel pathogens. Maintaining the absolute structural integrity of this microscopic seal is a non-negotiable prerequisite for human health. If this wall holds, you thrive. If it fails, disease follows. Yet modern life wages a constant assault on this barrier. Diets heavy in ultra-processed ingredients and high fat contents, compounded by chronic psychological stress, bombard the epithelial lining. These environmental stressors systematically wipe out the beneficial bacteria living along the gut wall. In their absence, harmful opportunistic microbes known as pathobionts rapidly multiply to fill the void. This state of microbial imbalance is called dysbiosis. As these harmful bacteria colonize the area, they actively consume the thick layer of protective mucus that normally coats the intestinal wall. Without that chemical buffer, the tight junctions are left fully exposed to a hostile microscopic environment. Dysbiosis effectively primes the gut for structural failure. This microbial shift creates a volatile environment where the once stable defensive wall becomes an active liability. That structural breach is initiated by a specific protein called zonulin. Under normal conditions, zonulin acts with precision, briefly loosening the tight junctions between intestinal cells to allow controlled transport, then restoring the seal. It is a finely tuned mechanism essential for balance, but when the microbiome is disrupted, this precision is lost. Zonulin is released in excess, and what was meant to be temporary becomes prolonged. Zonulin binds to receptors on the surface of intestinal cells, activating the EGF receptor, an internal signaling switch. This triggers a cascade within the cell, leading to the gradual disassembly of the tight junction proteins that hold the barrier intact. Piece by piece, the structural integrity weakens. The once tightly sealed connections begin to loosen and microscopic gaps start to form. Through these newly opened pathways, substances that are meant to remain confined within the gut, bacterial fragments, toxins, and partially digested food particles begin to seep into the surrounding tissue. The immune system, ever vigilant, recognizes these as threats. It responds swiftly, initiating an inflammatory cascade. This is leaky gut, not a result of external injury, but a consequence of internal dysregulation. A native protein performing its intended function, yet pushed beyond its physiological limits, quietly, systematically unlocking the very barrier it was designed to protect. Among the debris pouring through these open gates are Lipopolysaccharides or LPS. These are dark, jagged fragments of bacterial cell walls that drift down through the tissue and plunge straight into the systemic bloodstream. To the human immune system, LPS is a potent endotoxin. Our biology is hardwired to recognize these specific bacterial signatures as a lethal threat, requiring an immediate aggressive counterattack. As these toxins circulate, they inevitably collide with toll-like receptors, specifically TLR4, which sit on the surface of circulating white blood cells, like biological tripwires. That collision triggers an instant chemical cascade. The activated immune cells flood the bloodstream with reactive proteins called pro-inflammatory cytokines, placing the heart, the liver, and the brain on maximum alert. What began as a localized disturbance in the digestive tract has now bypassed containment. The barrier breach has amplified a microscopic bacterial shift into a systemic biological fire. When the gut leaks continuously, this immune alarm never shuts off. The body settles into a state of meta-inflammation, a chronic low-grade systemic inflammation. That becomes your new biological baseline. This data visualization shows how that baseline impacts the body over time. It plots the systemic accumulation of a microbial metabolite called phenylacetic acid or PAA directly against a rising curve of arterial stiffness. As PAA leaks from the gut and travels into the circulatory system, the arteries demonstrably harden. PAA induces cellular senescence in your blood vessels. The endothelial cells lining your arteries stop dividing and begin secreting their own inflammatory molecules, forcing the vessel walls to begin thickening and stiffening, accelerating their biological aging. When the gut leaks, it does more than activate immune cells. It confuses them. Microbes or their metabolites can trigger T cell responses against self-antigens through molecular mimicry, where the immune system mistakes your own tissue for a foreign invader. These activated cells then migrate to the synovium, fueling the cycle of joint destruction seen in rheumatoid arthritis. Cardiovascular decay, joint degradation, and metabolic dysfunction often present as completely disconnected issues. Yet, tracing them backward reveals they share the exact same origin point, a compromised intestinal wall. Treating the distant symptoms of these diseases without addressing the gut is like mopping the floor while the sink is still overflowing. Medicine is now trying to address the upstream structural failure itself. Restoration depends on the production of short-chain fatty acids, primarily a compound called butyrate. Butyrate is a versatile metabolic byproduct naturally manufactured by a healthy, balanced microbiome. It functions as a biological conditioning agent. As butyrate coats the epithelial lining, it down-regulates local inflammation and sends chemical commands instructing the Claudin and Occludin proteins to reassemble the tight junctions. What is most empowering is that we can intentionally cultivate this repair process by consuming targeted prebiotic fibers and precision probiotics. These interventions act as specific fertilizer, fueling the exact strains of bacteria required to mass produce these healing short-chain fatty acids. Sealing the gut barrier shuts off the flow of toxins into the bloodstream, removing the primary fuel source for systemic inflammation. It is a foundational strategy to recalibrate our biological baseline and meaningfully extend human health span.